Hello, everyone. Welcome to the session 4 of the International Symposium on Biological Science. I'm Jacqueline Alves Leite. I am a professor at the Federal University of Goiás, and it's my pleasure and honor to introduce you to Dr. Lykke-Hartmann, Dr. Karin Lykke-Hartmann. Dr. Karin is associate professor in the Department of Biomedicine at Aarhus University in Denmark, and she is investigating how dormant follicles are activated. And she also is researching new strategies associated to reproduction and ovarian follicle development. Dr. Karin is an expert in genetic manipulation and pioneer in the development of 2 mouse models related to disease that affect the function of sodium-potassium ATPase, such as alternating childhood hemiplegia and familial hemiplegic migraine that are associated with mutation in the alpha-3 and alpha-2 subunit of sodium-potassium ATPase, respectively. Thank you, Dr. Karin, for accepting our invitation. We are looking forward to hear from you about female reproduction. So feel free to start your talk, and please take your time.
Thank you very much for this kind introduction, Jacqueline. It's really great to see you again, and I'm really pleased we had this pass going on the sodium-potassium pump and on the mouse modeling. But I will not be talking about this project today. As you know, my life has already had a little female reproduction in it, and I will be talking about that today. So, can you see my screen? Here we go. Yes.
Yes.
You see it?
Yes, we can see your screen and see you and hear you. Perfect.
All right. Well, so again, thank you for inviting me to talk about this female reproduction, which is a project very close to my heart. And as Jacqueline already said, my name is Karin Lykke-Hartmann. I'm professor at the Department of Biomedicine here at Aarhus University in Denmark. And to all of you, please feel free to reach out to me after this presentation. So first, I want to give a short introduction to reproductive endocrinology that regulate ovarian follicle and development. During the fertile age, the gonadotrophin-releasing hormone is a master hormone regulating reproduction and is released in a positive manner from the hypothalamus. It then transports through the portal system to reach the anterior pituitary, where it binds the receptors on the gonadotrophin cells. Once it binds the receptor in these cells, it starts a cascade which will synthesize and release the luteinizing hormone LH and the follicle stimulating hormone FSH. So I will use LH and FSH to refer to these hormones. And the receptors for these hormones are found on the ovarian cells, in the theca cells and the granulosa cells down here on the follicles. And this trigger a lot of signaling, which will end up producing the progesterone and also the androgens, and also importantly in this manner the estrogen. And the estrogen and these sex hormones, they stimulate the follicle development and also the maturation of these follicles that go through this ovarian cycle, and then finally you will have the release of a mature egg. Also, it's quite established how the progesterone and the estrogen exert the negative feedback to keep this loop regulated. So this is kind of the extremely basic of reproductive endocrinology in the female system. So when you look at the ovarian follicle development, which is also referred to as the folliculogenesis, it can actually be divided into 2 phases. If you look over here, you have this pool of primordial follicles that constitutes the female capacity for fertility, and it, on a monthly basis, a little cohort of these primordial follicles are activated and turn into primary follicles. And they can stay dormant for many, many years before they actually get activated. Then they start this competitive journey of growing and maturation, and only one will end up to be the one that releases a mature egg, as is shown here. And this whole ovarian cycle can be divided into a gonadotrophin independent and dependent phase. And this is quite important because when you look at the treatment offered today to infertile patients who suffer from some kind of ovarian dysfunction, they get only one treatment. And this is a standard hormone treatment which only helps those follicles that are responsive to hormones, and that is from the secondary stages. And this is actually what prompted our project initially, is to look at, what do these follicles actually respond to if they don't respond to hormones. And this actually was sort of, in a very brief frame here, what made us start looking at that. And if you look into the primordial follicles and the more into the signaling, it's already known that in dormant follicles, this is what this off refers to, that is not activated. You have the P10, which is one of the gatekeeper of this dormancy that keeps this pathway inactive. Once you get a stimulation from the KIT ligand, which is secreted from the surrounding pregranulosa cells, you activate this pathway and you will end up having a phosphorylation of the ACT. This phosphorylated ACT has several sort of activation steps. It can remove the inhibitory effect of the TSC1 and 2, which releases the TOR complex to be active, and it also can travel into the nucleus and remove P27 and FOXO3A, which exerts some inhibitory effects on the growth. And these are kind of known, but still we were knowing from many other manipulation assays that there must be other players that are important in this stage. And this was actually sort of a very simple question. What else is happening within the primordial follicle that would allow us to regulate them? So we started this some years back, and we got a lot of tissues from the hospital, and we cut it into pieces, and then we put them into slides, and then we laser capture them using this LCM machine. So basically what it does is that you stage the primordial follicle or the primary follicle extremely careful. And as is shown over here in a more schematic view, you can see that there's quite some significant difference between these two stages. So the primordial has these very flat pregranulosa cells encapsulating the oocyte, whereas the primary has this more cubic granulosa cells, and also the oocyte has grown in size. So we started collecting all these from different patients that we got tissue from, and then we asked what kind of molecular markers do they express. So we did this, and then we started doing RNA sequencing on these very distinct pools of either the oocyte or the whole follicle, including the granulosa cells. So this is what it looks like. So when you look at the images taken from the laser capture microscope, they are not the best because this is a microscope that has been designed to laser capture them. But here you see an example of a primordial follicle. Then you encircle them by the laser using the program, and then afterwards you collect your sample. And then this was done really careful to discriminate between the primordial and the primary. And then one of a student started to collect these, from different patients with a lot of different numbers. You can see, several hundred for some of the samples were collected this way. And then we sequenced them using 12 different libraries, and we started mapping them, and then we started annotating them into the human reference genome, and then we normalized them and transferred them into the log2 values. And then we started to look at what is going on in these cells, and in the dormant cell, are there anything that's really could pinpoint what kind of signaling we could communicate with these dormant follicles. And here is just a little bit, the first part is just a little bit of the summers, how we annotated them, mapping them and filtered them, and then we started to look into what is actually significantly expressed in these different single cell compartments, so that what we did was to make sure that there was no differences between the patients, but they were all the same. And then we started to look at how many genes would fulfill these criteria, and then we started to look at what is exactly the difference between for instance, here for the granulosa cells that are surrounding the primordial to the granulosa cells when they are surrounding the primary cells. And then we just started to do all these different kind of analysis, up-regulated, down-regulated from the granulosa cells, all the oocytes as well. And then we found all the components and we tried to put them into signaling pathways, figuring out what kind of signaling pathways are actually active once you start to transit from the primordial to the primary follicles. And then we found a lot of different hits. That's what you always do when you make these global analysis. You're always finding a lot of interesting things. And then we thought, okay, let's do some functional testing about this and set up some kind of pharmacological testing of the pathways using an in vitro based system. So we use mouse mostly as our model system. We took out the ovaries, put them into inserts, grow them with or without a drug that could hit a specific component of a pathway that would either work as an agonist or an antagonist. And then after some titration with time and concentration, we sliced the ovaries and then we just started really counting, what kind of effect could this drug have on how this first initial step in folliculogenesis was regulated. And we just counted either the primordial and the primary and secondary follicles. And we only counted follicles where the nucleus was visible to ensure that we didn't count them more than once. And then we just started this quite systematic drug in the beginning, testing of the drugs in the beginning, just titration in time and concentration. And we ended up with some extremely interesting targets of how we could actually communicate with these dormant follicles. And I'm just going to show a little bit example of how we assess what's happening once we start to manipulate these follicles, which became really interesting. So one of the things that we do is that we treat them and then we look for apoptosis. Are there any increased apoptosis that kind of suggest that it's not really good for the cell? And then we also look at the diameters, because some of the drugs that we tested for different pathways were so powerful that some of the oocyte almost blew up, and then of course it's not really good for the cell. We also looked at mitochondrial membrane potential to see if there was any problems in the energy sources or the oxidative species, the reactive oxidative species, so that would sort of indicate that there was any oxidative stress going on. And we found for some of the drugs and some of the pathways, this was not compatible with a very good oocyte quality, and that would never, potentially be able to be translated into a clinical use. But then we have some drugs that actually showed that they were actually compatible with an extremely high quality of the egg. So we then, after we have been treating some of the follicles, we also went on to sequence single cells from these using the same approach, and as I showed before, to see what kind of molecular changes could we put onto the cell, and are there indications that we have disturbed any hormonal interference. And some of them just showed a really, really nice and clean profile. So what the next was is, once we have been looking at all these parameters, they all look fine. We couldn't see, and then of course what is really important is to see that once you treated the eggs, you can actually get to produce a litter out of it. And we did that in different ways, and I'm just gonna not spend that much time on this, but just to show that we took out the ovaries, put them in the culture with the drug or without the drug. Then we grow them as they come bigger in the follicle size. They need a different support, and then we grow them in a different media setup to make sure they can develop these granulosa cells. And then once they reach the enteral part, we hormone treat them to form this mature egg. This is a metaphase egg. Then we did an IVF for them to see that they could actually be fertile after being treated with the drug. And then we some of those that we did for IVF, we put into the incubator, and we followed their growth all the way up during the pre-implantation stage to the blastocyst stage. And some of them we took at the 2-cell state and transferred back into a female recipient to see if they could also generate leaders after this treatment. And this was really nice to see that it was possible, and we got a really nice result having this. And we also did it with some really old mice, which will mimic some of these patient that could eventually benefit from a new treatment of this. And we saw that when we use this, some of these drugs, we actually get a significant increase in generating mature eggs. We also looked at the morphology of these blastocysts, and they seem to be the same in both groups. We went back and tested it on some tissue we got from patients, and we can see here that there are some sections here that shows the human tissue, which is a little bit different from the mouse tissue. And when we treat them with different drugs, we can actually see that we can enhance the activation of the primordial follicles. So now this tissue we got from different patients that are infertile from the hospital, and they're still infertile, but we definitely see the clinical perspective in developing this method, because now we can generate follicles from this tissue. So this all in all has been quite a nice journey for us to go through this stage. And just because time is running, I can see I will go through the summary and perspective, just to say that we have identified pathways that can regulate this first hormone independent transition, going from primordial to primary transition. We also did some functional testing to show that we could actually target different kind of pathways to enhance this activation, and didn't really affect the egg quality. Some of them did, but not all of them. And we could also generate litters in vivo, and it also was translating back to human tissue that we got from infertile patients. And now we are addressing different pathways to see what are the mechanism of action for these drugs, and how does that translate into something that could eventually move forward for a new infertility treatment. So I'll just end up with thanking the team at Aarhus University, Department of Biomedicine. Some of the key players are listed over here. We have Mahmoubea Mushahi, who is an assistant professor in the lab, Erik Ernst and Emil Hagen, who are the medical doctors helping to collect the tissue from the hospital. And then we also have Anders, who is a lab manager. And of course, we have a lot of students, PhD students, medical students, and master students who are helping with all kind of testing of these projects. So that was it for me. So thank you very much, and I'm happy to take questions, or please feel free to follow up afterwards as well. Yeah.
Thank you, Karin, for your wonderful talk. It's always nice to learn with you about science, about different talks in the science. It's too new for me, okay, but
I know this is
nice
new, and it has nothing to do with the project.
You.
Yeah.
I have one question.
Sure.
Yeah, I have.
Sorry.
Karin?
Yes.
Sorry, I think we have some connection problem.
Yes, I hear you now.
Yeah, I can see you and hear you perfect now. Okay. So I just say that it's a little bit different for me, because it's completely different that they are area that we work together. Yeah, but it's so interesting area, and I think we can try to collaborate with this topic.
Yes, that would be really
as
nice.
soon as possible.
Yeah, that would be really nice.
So, but I have one question about this, because
Hi, Professor Karin. Sorry, we are having some connection problems.
Yeah.
Let's see if Jacqueline can deal with this problem. Just a
I
minute,
heard the first
please.
part and then it was kind of lost.
Yeah.
Well, that's okay. So I know that it's always a challenge with these things.
Okay.
Yeah.
I'm going to see, I can see some questions here, Professor Karin. So before that, thank you for this beautiful presentation, Karin. Very interesting. So let me see here. So we have a question from Pericles Mesquita. He thanks for the lecture, and he says, I would like to ask if the oxidative stress is associated with the infertility.
Well, there's a lot of different theories about this. This is why we're really careful to try and measure some of these parameters, because it has been shown that it can affect infertility if you are going through a lot of oxidative stress. But mostly these are quite age-related, when the repair mechanism in the eggs are reducing themselves to go through this. And I think a lot of effort has been tried to go through this to kind of treat infertility, but it has not really been the way to go forward. So it has been, and it's a really good question, because a lot of people have tried to really target a lot of different things, also going through this ACT pathway and the P10 pathway, which I started to show, but it actually turns out to really put a lot of stress on the follicle, and especially manipulating a bit with the DNA quality, so that we found that the repair mechanism has been shown not to be function very well once you go into these kind of different drugs. So it is a really, really fine balance that you have to find here, and oxidative stress is truly a concern in this. Yeah.
Okay. Ericles also asks, and the use of natural products with potential antioxidant could help to treat infertility?
Well, some people have tried that, but I think because they are so all over you, I mean, they're not a specific target, right? So they're a little bit, potentially targeting a lot of things. It has not really been shown to be very effective, not to my knowledge, at least.
Okay. Jacqueline is back. Go ahead, Jacqueline. It's up to you.
Thank you, Professor Karin. Thank you, Professor Manoel. I'm sorry, I have some problem with my internet here. But I have one question, because several diseases have a chronic inflammatory, and I just want to know if this chronic inflammatory that present in this disease, like obesity or hypertension, could be do some influence in follicle development or follicle dynamics.
So, well, actually, I skipped the slide showing a little bit of different side of disease coming into the infertility because, of the time. But I think if you are in the obesity category, your follicle development is sort of affected in a different way. Many of these patients will also suffer from PCOS, which is quite a different disease. Our aim is to go more for the low ovarian reserve patient, those who are either premature or age-related, because then you do not really benefit from the hormone treatment today, because either you don't have any secondary follicles because your reserve is low, or you don't really tolerate the treatment because you already have endogenous levels of FSAs that are too high for you to tolerate it. When you have obesity, it's a different problem, and that's going through a little bit in the insulin that you have this insulin resistance, and it also affects a bit the way that you synthesize the androgen pathway. So you get much more sort of these androgen into your system, so the phenotype will be slightly different. You do have a lot of follicles and you can actually activate it, but they do not develop. Right, they get into these more cystic. If you go into the inflammation, it's a really different area, and it's a huge area. And we also tried to look slightly on this, and we looked at some of the toll receptors because they're quite highly expressed. And we thought, either it's just because it's ready to combat, or because that it's wanting to do something else, and we could not really figure out. But it definitely is ready to combat. So if we challenge the oocyte with some antigens, it definitely is alert immediately. And if you have these inflammatory diseases, it might be that you kind of stop the activation a bit, not because you have low reserve, but because your system is taken over by these inflammatory pathways. So there are a little bit different sort of etiologies to how you become infertile. So it's some really complex areas, and especially when you come to PCOS, because the endocrinology is a little bit complex with the androgens. But yeah, quite exciting.
Okay, it's really exciting to study more about this topic.
Yeah, I think, and it will also be relevant for the systems eventually, like everything else in the body, right? It can translate, but you just find the system to look in. Yeah.
Exactly. We have a lot of questions here. I think we have more time. So the questions, the second questions comes from Ellen Christina. Ellen asked if maybe she missed it, but did your group also proceed to in vitro culture of human follicles or on rodent?
Yeah, well, we do process human tissue in the lab now. And I know this is another extremely big area in the fertility world, because the clinical perspective in being able to mature human follicles in vitro is huge. So the IVM protocols are definitely something that is on many people's list of things to do and accomplish. And it's extremely tricky because the rodent has been done for years, and it can be done, with a few techniques, but it doesn't really translate very well with the human tissue, and it's extremely difficult. And of course, it comes to the end, you cannot really test the quality of the eggs because you're not allowed to do IVF just to check it for the humans with, for ethical reasons. But it's an extremely difficult protocol, but we do it. Yeah.
Thank you, Karin. The next question is also from Eileen. She asked, also, are human samples obtained from healthy patients, or was there any disease associated to the samples? And she said that thank you very much for your lectures.
You're welcome. So So when we started this project, trying to look into the molecular maps of these primordial and primary follicles, we took it from healthy patients. So patients who did not have any diseases related to the ovaries. So most of them had their ovaries removed because they were going into chemotherapy for another cancer disease. We cannot, of course, not exclude, that there might be something, but on the ovary side, they look normal. So once they have been cured, so what you do is you take out the ovaries, you cryopreserve them. Once a female has been cured for the cancer, we can actually put the ovaries back into her, and then she will actually start cycling. So it's really amazing. And it was one of the procedures that was actually pioneered here at Aarhus University Hospital some years back. And some of the first women who had their ovaries transplanted back after being cured from their cancer has already had children. So that's quite significant. Once we started to test some of the theories we had on these things, different things, they're not published yet. So we are just checking up a few things. We took the tissue from infertile patients suffering from the premature ovarian insufficiency, POI patients, to see that it could have an effect of those who have a really low ovarian reserve. Yes.
Thank you, Karin. I think we have more time for one question. The next question comes from Paulo Guedini. He asked if you saw some markers of oxidative stress in the experiment.
Yeah, I only showed some of the analysis we did, and we did a lot of different analysis. Some of the sort of more general ones that we have used was to look at the mitochondrial membrane potential. It actually says if the mitochondria start to get stressed, and you will immediately see a difference in this membrane potential. We also used the Ross assay, as you know, for reactive oxygen species, to see if there was any differences in this. And then, of course, we look at tunnel assay, which will just show you if there's any increased apoptosis, because if this is going to be eventually translated latenable into a fertility treatment, you want the egg to be superb, right? You don't want them to have any apoptosis. We also looked at the BCL2 and the box markers I didn't show you here. And we have also been sequencing, RNA sequencing, all of the stages throughout the development, single cell from RNA sequencing. And this is what we are analyzing right now, and that will tell you really in details what's going on in each cell after they have been treated. So we are not quite finished that analysis yet. It's actually still ongoing, and some of them we are repeating now. We had a laser that broke from the microscope. You all know that these technical problems always happens when you don't want them to happen. So we had to fix the laser, and now we are collecting the last cells. And it's always like this when you do science, right? So for us, it's really, really important that we have all these quality markers, ticked off, combined with more functional data to show what's going on. So yes.
Thank you, Karin. I think we have time for one more question. The questions comes from Ericles, and he asked about the association of environment pollution and future infertility, and how to protect against this kind of stress once we have a lot of toxic agents such as aluminum.
Yeah, I mean, this is a really good question, and it's a lot in the, I think it's a lot in the news lately, right? With all these, potential pollutions that can affect the fertility, and that goes for male and for female, both of us. The sexes who are constantly being exposed to all kind of pollutions and so on. And we also see there's a great increase in infertility in both sexes because of this, all the plastic, hormone disturbing drugs, and toxic agents, aluminium, and all these kind of things. I don't know how we protect ourselves from that. I really don't know. I think we really have to be conscious about how we live lives in general, and how you take care of yourself. So I don't really have a solution to that, and I know many people are doing a lot of this research. So, and I can just see the last question of Ellen, who came back with this from Telfos group. I don't know if you have tried that protocol, but if you have, I think this protocol needs a lot of optimization, because I know that group very well, of course, and I also know of her work, but the protocol needs a lot of optimization to be robust. Yeah.
Okay. Thank you, Karin. It's a really good question that Ericles do. Manoel, Manoel, I don't know if you have some time for one question.
Yes, Jacqueline, we have time for questions. Go ahead, Jacqueline.
I think I think the next questions, I don't know if it's from Ellen.
Yeah, but I took it a little bit. Yeah. Because I thought you said you only have time for one more. I just, I can see she's answering also. I just wanted to make sure that, it's not like it has not there's one protocol being published, but it's not a very good protocol. I'm really happy to know if it's working for you, but for sure it's not working for us. That protocol is a little bit, needs some optimization. If you want to phrase it that way. We are definitely making our own protocol at the moment for this, for the human tissue. Yeah.
Okay, it's just a comment that.
Yeah, exactly. No, I just want to make sure, because often you think if something is published, it's going to work fine, the problem is solved. It's far from the case, right? In many cases, maybe it worked once, this is not a very efficient protocol.
Yeah. Okay, so I think we don't have more time for questions. So thank you, Karin, for your joy with us this morning.
Of course.
It was really nice to see you again.
I hope I can soon come to visit you again.
Yeah, I also hope.
Yeah.
I hope to visit Aarhus University again, and maybe being in your lab for some time. Thank you for your wonderful talk, And if you want to join with us here, just be here.
Okay.
The next, so we will move for the next talk, and the next talk will be coordinated by Professor Manoel. Thank you, everyone.
Thank you, Professor Jacqueline. And before starting our next presentation, I'd like to thank you again, Professor Karin, for this beautiful, amazing presentation and for the very interesting discussion about this topic. Thank you again, Karin. So let's go to our next section, our next presentation, who is going to be delivered by Professor Claudio Barbeito. First of all, Professor, I'd like to thank you very much for joining us during our International Symposium on Biological Sciences. Professor Claudio Barbeito is PhD in Veterinary Sciences. He's Professor of Histology and Embryology and General Pathology in the School of Veterinary Sciences in National University of La Plata, Argentina. He is a principal investigator of National Council for Scientific Research and Technological Extension. He also has 167 papers indexed in Scopus about histology, pathology, and development of vertebrates, mainly reproduction aspects. He also has 25 supervised doctoral theses. Professor Claudio, again, thank you very much for being so kind and for having accepted the invitation to be here. It's an honor having you here. So feel free to start your lecture when you want. Thank you very much again.
Thank you. Good morning. I would like to thank the organizer for this kind invitation. It's a pleasure to be here. Today I will talk about the plains viscachas Lagostomus maximus, female prostate. In particular, I will discuss what is different and what is shared when compared to other mammals. I have been working on this topic for some years ago, together with Alicia Flamini and Enrique Portiansky in the University of La Plata in Argentina. So, who are the plain viscacha? They belong to Stricognathi group. They are rodents similar to capybaras and guinea pigs that live in communal burrows systems in groups known as viscacheras. Here is Lagostomus maximus. They have some unique reproductive features that are not present in other Eutherian mammals. They have polyovulation and produce around 200 to 800 oocytes. From them, only 10 to 12 finally result in implantation, and only 2 pups are born from them. These successful embryos are those placed in the most caudal position in each uterine horn. The rest of them are reabsorbed in some moment between gestational day 26 and 70. So, how did we were interested in the female prostate of these animals. Some years ago, Alicia Flamini was working on her PhD thesis under my supervision. She was interested in the reproductive system of Lagostomus maximus. One day, we were at the lab, and she called me to show me something under the microscope. This image. She asked me to tell her what it was. Immediately, I said, it's a prostate. And so she moved to another part of the same slide and showed me this. And what is this? Well, it seemed to be a vagina. So we were surprised, of course. We had read some mentions to a prostate gland in female, but never seen in real life. We revised all the samples we had, and in all females there was prostate. Amazing. So the next step was to carry out an extensive search for references. So, what we found in this search. In the beginning of the 20th century, Rauter observed the presence of a well-developed prostate in the rodent Arbicantis cinereus females. Some years later, Mark noted the existence of prostatic lobes in the female rat. By the time Dinley found this gland in some insectivores like the hedgehog. And Price and Mahoney showed that these glands arose at bottom the ventrolateral walls of the urogenital sinus. There were reports that in some species, such as dog, rudiments of this gland are occasionally found. But in any other Stricognati rodent, this gland was found up to here. When we expanded the search to note what happened in humans, we found that it was de Graaf who described the gland in humans for the first time and introduced the term corpus glandulosum or female prostate. The father of the pathology, Virchow, described female woman prostate as a genito-urinary organ. And also, he was the first to describe corpora amylaceae similar to those of the male prostate. In 1880, Skene identified in human two large ducts and named this structure Skene gland or ducts. This name is not longer used because, as you saw, he was not the first to describe human female prostate. Skene emphasized the role of this gland in some infections of the female reproductive systems and introduced the term skenitis. We are ready to come back to the 20th century, and in the 80s, Milan Saviasic started to study the gland in humans. Years and years of work give to his book, The Human Female Prostate. There, he stated from the human female prostate was a functional genitourinary organ. A Milan is a forensic pathologist that has some controversial ideas. For instance, that this gland corresponds to the G spot in women. He also carried out study on the chemical composition using histoenzymatic and immunohistochemistry techniques. These are photos that the studies I was talking about. He found that in the adenomers of the gland, they were cells expressing some molecules that are the same as in male. For instance, the acid phosphatase. It was found in humans at different ages. They also found reactivity for the prostate antigen. It was key in forensic science because some of these molecules were used as evidence of rape, since they are present in the pre-ejaculatory liquid. So, this finding forced to a revision of the use of them as evidence of rape by its own. Some later studies found that in some animals, such as the rat, the gland is constant and has similarity with the human female prostate, as well as with the male gland in the same species. In this study, the use of immunohistochemistry revealed the presence of prostatic-specific antigen and prostatic-specific phosphatase. So here you have some of our first contribution. We performed macroanatomical and histological description. Regarding histology, we use traditional techniques such as hematoxylin angiosin, trichromic techniques, silver staining, or cyan staining to observe elastic fibers, and other techniques such as us. We found that in the plain viscacha, the prostate is placed around the urethra. This is urethra. This is prostate gland. Urethra, ducts of the gland, adenomers of the gland. It has a lobular pattern. We can see the different lobes, lobule. Is formed by alveolar adenomers with a secretory aspect, which is confirmed by the presence of secretion in the lumen. There are also corpora amylaceae, similar to those found in the male and female of other species. Finally, the ducts finished in the urethra in two main ducts that go independently to the urethra. In the first year of the 21st century, the group of Dr. Taboga, in which Professor Manoel participates, started studies on female prostate of Charville's Meriones ungiculatus, other muridae rodents as a rat with a constant female prostate. They demonstrated that also in this species there are morphological and biochemical similarities between male and female prostate. Also, they found an effect of strong progesterone, testosterone on the gland. And also they found an action of endocrine disruptor as the bisphenol A. So this group also studied some aspect of prostate development and found homology between female prostate and the ventral lobe in the male. Finally, they also found that pregnancy produced change in the gland related to an increase in seric progesterone. So coming back to our work, after the first result, for some years, we left the study of female prostate of the viscacha to dedicate to other organs of this animal, the placenta, for instance. And then we came back to the prostate. We applied not only technique of traditional optic microscopy, but also lectin histochemistry, immunohistochemistry, and electron microscopy. Our aim was to describe more deeply this gland and to compare pregnant and not pregnant females. So we found that pregnant and not pregnant female have a similar aspect when observed in the optic microscope. Here are pregnant, here non-pregnant. Also, we corroborate that in the stroma, there are smooth muscle fiber as observed using antibody anti-desmin. This was found in pregnant and not pregnant female. Positive nuclei were found detecting PCNA as a marker of proliferation cellular. Here in the pregnant group, we found more nuclei positive to PCNA. Here, there are pregnant, there are non-pregnant. Using the Picosirius technique, we found the Picosirius to determine collagen fibers. We found that in pregnant and not pregnant females, the stroma has mainly type 1, pregnant, not pregnant, type 1, and to lesser type 3, is difficult to see, in a similar amount in both groups. We used lectin histochemistry to detect a specific carbohydrates that are present in glycoproteins. Here was a clear difference between pregnant and not pregnant females with a stronger, more scented and varied staining in the pregnant females. Using scanning electron microscopy, observe epithelial projection, here in pregnant, in non-pregnant, with a connective axis that form the adenomers of this gland. This observation was shared by the two groups. Also with transmission electronic microscopy, we found cells with a secretory aspect in pregnant and no pregnant. You can see the granules. There was abundant rough reticulum, endoplasmic reticulum here, for example, and a Golgi complex here in both groups. We also found some clear cells that could be intermediate cells described also in humans and that were interpreted as a transition between basal and tall cells. Another thing is a confirmation that the epithelium is pseudo-stratified epithelium with basal cells that are the positive to PCNA. There are the basal cells. Well, to conclude, we found that the female prostate of the viscacha shared some features with other species. We found a pseudostratified epithelium with basal cells with a proliferative activity. As in the male, the female has smooth muscle tissue in the stroma. Tall cells have ultrastructural and histochemical characteristics that indicate secretory activity increases during pregnancy. Our work leaves some perspective to continue. For instance, something interesting is that we did not find reactivity to prostatic antigen, something that is common in other species. But it's important to remark that in male prostate of Lagostomus maximus, we did find reactivity in viscacha specimens. Another thing, how is development in Lagostomus maximus? Is there homology between ventral lobe of the male and female prostate also in this species? Another question. Up to here, the functionality of this gland is unknown for female mammals, if it has any function. In gerbills, it was found a stimulant effect of progesterone. It is interesting because the viscachas during pregnancy have several corporal lutea. In fact, they have a process of pseudo-ovulation during the pregnancy that increases more the number of corporal lutea. In the future, it will be interesting to study the link between progesterone levels and change in secretory cells in the female prostate. Well, this is the group that work with the viscacha's prostate. Enrique Portiansky, Alicia Flamini, she was your doctoral thesis about the reproductive system of the viscacha. Eduardo Jimeno in the initial studies. Alcira Diaz performed the histochemical study in the last paper, and Francisco Acuña, he is continuing the research about this gland. Thank you, and excuse me by my English that is not well, is bad.
Thank you very much, Professor Claudio. Please don't worry about English language. And I know I'm a very suspect to say that your lecture was very interesting, and it was, of course, very interesting and very beautiful, because I work with female prostate, you know, very well. And also, Professor Claudio, very beautiful, your images from female prostate tissue using different techniques, very beautiful. So we have time for questions. Let's see what we have in our chat. So, okay. Fernanda Cristina, Professor Fernanda Cristina Alcântara dos Santos is asking. First, he says, thank you very much for the lecture, Dr. Claudio Barbeito. The question is, does plain viscacha develop lesions in female prostate as well as other rodents?
Yes. Yes. Thank you, Fernanda. We don't found lesion in the female prostate. We saw 100 prostate of viscacha, female viscacha, and any lesion was found.
Very interesting, Professor
but
Claudio.
These animals are captured, notar in bioterio. No son animales criados para el trabajo, entonces son capturados y nunca en alrededor de 100 animales hemos encontrado lesiones.
Very interesting, Professor Claudio, because for gerbil is very different, because female gerbil it's very common to find lesions, especially in older animals. It's interesting. Professor Fernanda also asks, is plain viscacha a good model to access the effects of endocrine disruptors on the female prostate?
Bueno,
like bisphenol A?
The problem is very difficult criar viscachas en cautiverio. Es muy difícil poder criarlas en cautiverio because she lives in the viscacheras, in deeply covered, and una sola vez la profesora Flamini could have viscacha reproduction in cautiverio. Es muy difícil. Entonces, voy a tratar de contestarla en español. Para usarla como modelo de disruptor endocrino se complica mucho. uno debe tener la hembra en jaulas y en general no se preñan, is non pregnant in cage. Sería muy interesante, very interesting, because the viscacha have many other particularity in its reproduction, as the polyovulation, the resorption, and the effect of endocrine disruptor could be very interesting.
Thank you, Professor, for your answer. We have more questions. Edivaldo Mendes, he first thanks you for the excellent presentation. And he asks, do you have any hypotheses about the role of the female prostate in reproduction? This is a 1 million question.
No, could be lubrication or generate close to the vagina, but because it's the only rodent of the Hystricognathi group, because the rat have constant gland and mouse not have the gland because the chinchilla in the same family that the viscacha have not the gland. In the human, Sabiasic considered the possibility that the gland is a G spot, but in the animals, it is possible. But with lectin histochemistry, we found a great variety of carbohydrate and indicate a variety of glycoproteins, but the function could be very interesting. Make a proteomic study of the gland.
Yes. I would say that this question is the 1 million question for us that study female prostate. Okay, we have more questions from Jessica Souza. She says, this model appears to have more pleated epithelium, that enfoldings of the epithelium layer, than the model used by Professor Manoel. Is there a difference in gland secretion?
Yes. Yes, Jessica, one difference with the model of Professor Manoel is that them found that progesterone not stimulant the cell proliferation in the gland. And in viscacha, the pregnant female have more PCNA marker cells. Is a difference. And other difference, we cannot explain this, is the negativity to the prostatic specific antigen. In all the species, cervil, rat, human, the gland is positive as a male. In viscacha, is positive in the male, but not in the female. It's very strange.
Thank you, Professor. We have another question from our audience. Natane Ribeiro Lima. She thanks you a lot for the amazing lecture. Her question is, what's the function or what's the importance of the acid phosphatase in the female prostate?
No, it's not known because it's not known the function. Is could be that any function have and is a vestigial organ because there are a very interesting study in the vesicle in the vesicula biliar, bilis organ that bilis production that is present in that bilis have the bile bladder, vesicula biliar. Don't remember the name. That a gene is not expressed in the rat and the organ not developed. I think that in this case could be that an effect that inhibit the development of the gland in most females. In some species, the gene, I don't know which, is not explicit, and the gland develop as in the male. It could be that the gland have any function and is a vestigial organ.
Thank you, Professor. Professor Claudio, you told your group have analyzed 800 viscacha. It is a lot of animals. It is possible to find prostate in all females. I'm asking this because for Goebbels, it's easy to find female prostate. I would say that almost 100% of females have prostate, like we know that happens in women. But for example, if we try to do this using rats, I mean oyster rats, it's very complicated because most females of rats don't have prostate, you know. And about viscacha,
Is I don't not hear Manuel.
Sorry, Professor. I'm going to ask you again. Can you hear me?
Is the internet is complicated. I don't. It cut.
Okay.
Could,
Let me try another question. Can you hear me? Yes. Now?
No. Yes.
Okay, Professor. Did you perform measures of the epithelial height in the female prostate of that pregnancy to see if the epithelial layer is higher?
Is a future research. We return the prostate study because Alicia Flamini was performing your thesis about other the reproductive organs. We study the placenta and the embryo death and resorption, that is very interesting, and we published 4 papers in the last years, and now we are making new work about the prostate. We are designing experiment using morphometric techniques, comparing a different state, and we go to immunohistochemistry application of receptor to androgen and to progesterone to see change in pregnant and not pregnant.
Very, very nice.
And in
It's good to
different
know.
moments of the pregnancy.
Yes, yes. It's good to know about that, Professor. So, Professor Claudio, we do not have more questions. And in fact, you have to follow to the next section, to the next presentation, sorry,
Yeah.
of our section. So again, Professor, thank you very much for being here and for having contributed a lot for improving our understanding of the female prostate. Thank you very much. So now we are going to follow with our next presentation. So I'd like to introduce Professor Paul Perjesi. First of all, Professor, I'd like to thank you very much for being here and for having accepted the invitation to joining us during our International Symposium on Biological Sciences. Professor Paul Perjesi graduated in pharmacy from the University of Sweden. He bagged his PhD in pharmaceutical chemistry at Albert St. George University. He is a member of the Pharmaceutical Sciences Committee, Hungarian Academy of Sciences. He is a full professor at the Institute of Pharmaceutical Chemistry at the University of Pécs. He has published over 100 articles. He's an author of a book too, editor of 2 books, co-author of 9 book chapters, and an editor-in-chief for the Journal of Pharmaceutical and Biopharmaceutical Research. Professor Paul, thank you again very much for being here and for being so kind and having accepted the invitation to be here with us. It is an honor having you here. So feel free to start your lecture when you want. Thank you very much.
I do thank you very much for this very kind of invitation. First of all, in the introductory part, we were talking about my association with your university, which is fairly close on the field of reactions of chalcones with different thiol substances. However, all the 2 PhD students who graduated in my and Professor Paridat's joint tutoring has got very good results, continuously publishing, and I'm very, very how to say, it was my really great pleasure to have guest researchers from Brazil and keep this content alive. So thank you again for the invitation. My 2 days lecture, I do hope that it's visible soon.
Not yet, Professor.
Yet, but I think maybe I have to do something here. No? Maybe we have to wait for it. Should should I do something or my lecture is open. I mean, the PT is open now. It's on the screen. So I'm waiting for your colleague to share the screen. Okay.
Okay,
so,
Gustavo, can you help us?
Or just ask him to.
Gustavo is saying, Professor Paul, just open your PowerPoint.
Yes, it's open now.
It's open.
And start the.
Did you share your screen with us?
Yes.
Yes. Okay. Now it's gonna work.
Okay. So you can see the.
Yes, I can see your screen, but not your presentation.
Yeah, that's my problem. So it's on the screen, and
now it is okay, Professor.
Is okay now?
Yes, it is okay.
It was.
It was.
How about now?
Okay, nice.
It's okay now?
Yes, it is okay.
Yeah. So let's go.
Just, Professor, just put on full screen, please.
This way?
Not yet. Just put to show in full screen, entire screen.
Oh, yeah. Oh, okay.
Okay.
It is full screen now.
Yes, now it's perfect, Professor. Thank you.
So we can go.
Yes.
Okay, thank you so much. So, as in my introductory discussion with Manoel, Professor Manoel, I was talking about a sort of flavonoids, chalcones, which are open chain precursors of this important natural product. And this lecture concentrates on the potential use of chalcone derivatives as antiviral, anti-SARS in particular agents. Let's start my lecture with some statistics. If we have a look at this figure, we can see some important figures of the coronavirus pandemic. First of all, I just mentioned two figures. So the global cases in May was 156 million, and the global death was, let's say, 3.3 million people. And according to the 2 days figures, both cases went up statistically different way. So the total cases now is 250 million altogether, and the total death is 5 million people. It's a lot. It's a huge number. It's unbelievable in the beginning of the 21st century, but this is the case. Now, at the beginning of my lecture, I wish to tell some details of the virus. We do not have enough time to discuss all the details, so I will concentrate only those particulars which are related to the Chalcones' possible antiviral applications. The classical classification of viruses, what I've learned when I was pharmacy student a long time ago, is like this. Namely, we can distinguish DNA viruses and RNA viruses, and among the RNA viruses, there are different two different classes in RNA-dependent RNA polymerase or with reverse transcriptase activities. Coronaviruses belong to this last one. It's I think is well known now all over the world. And this is a very simplified structure of the compound, just recalling some important proteins and which plays important role in the development of the disease, and of course plays important role, represent important targets of possible antiviral therapy. So it's a single-strain positive sense RNA is the genetic material of the virus, which is associated with the nucleoprotein and the glycoprotein, the so-called spike protein, and sometimes ham agglutinin esterase proteins are also found in some sort of SARS and MARS viruses. A new class of classification of viruses is the so-called Baltimore scheme, which distinguishes 7 different classes of viruses based on the more precisely specified class of the genetic material. And among this one, we will find the coronaviruses as among the single-strain RNA viruses. Let's see what does it mean. The positive sense single-strand RNA, which is the viral genome of the coronaviruses, can use their genome directly as a messenger. So it means that as soon as this genetic material is released in the host cell, it can act as a code for translation by the host ribosome to synthesize the necessary viral proteins, and as for replication of the genetic material of the virus, it uses RNA-dependent RNA polymerase or RNA replicase, which creates a negative strand RNA, and this is converted to the positive strand RNA by the RNA-dependent RNA polymerase again, and that way both the viral protein and both the genetic material can be replicated. So before we wish to go into details of the antiviral therapy, I wish to tell some words about the immunization, which is obviously the most effective antiviral therapy at that moment. And the reason why I go up with these few slides incorporated into my lecture is that one of the most successful line of immunization has got a Hungarian link. Now, briefly, all the vaccines that are used nowadays are effects or produce the so-called active immunization, and this different kind of vaccines stimulate proliferation of the T and the B cells, resulting formation of effector, producing antibodies, and memory B cells, as it's known as the basic immunology. And the formation of these memory cells is the basis for the relative permanent effects of vaccination, which is under question now, and actually we are in the developmental phase concerning this sort of this aspect of this immunization. There are a lot of different vaccines developed so far earlier not to mention those ones which are particularly developed to obtain immunity against the coronavirus too. In this lecture, I wish only to tell some words about the messenger RNA vaccines. The messenger RNA vaccine is, to the best of my knowledge and to my opinion, is the most successful vaccination at this moment. The messenger RNA vaccines make protein, the spike protein, and to trigger immune response. But what was the difficulties of this project? There were a lot. One of the difficulty is the well-known instability of the RNA. This instability was overcome by a simple chemical transformation of the uridine units of the coded RNA to the so-called pseudo-uridine units. These two compounds only differ in the linking position of the ribose unit to the heterocyclic ring, namely in the natural uridine. This is an N-glycosidyl linkage, which makes its instability, which can be improved by eliminating this 2-hydroxyl. However, in pseudo-uridine, as we can see, the uracil unit is linked to the ribose unit through a carbon-carbon bond. That was one of the inventions, one of the most important inventions that the two scientists, Dr. Garrido and Dr. Weissman, developed. And there is a very good recent review in Nature about the other details of this development. The other important feature is the lipid layer of the nanoparticles, which into which this RNA is incorporated. This lipid layer consists of 4 different lipids. Among them, the ionizable lipid part plays a very important role. So, as a result of these two improvement findings, it could obtain a useful medicine right now, which can get into the cells, keeping the RNA stable and not to recognize the macromolecule as antigenic. So these two, as a result of these two development, the circle, you are very much familiar, so I do not repeat all the details now, is working, and the vaccine is safe, and to the best of my knowledge, these two physical-chemical development could result in a successful drug development project. As for the anti-SARS-CoV-2 strategies, it's also a very wide topic. So obviously, I will just pick up one segment of the different strategies, which are related to the targets that can be attacked by Chalcones. So if we look at the simplified mechanism of replication of viruses that distinguishes the 6 more important steps, you might know that almost all the important steps can be inhibited by different chemicals, different drugs in different viruses. So, for example, the entrance inhibitors involving the ICA enzyme, the surface of the lung cells, for example, is also an important feature. We heard about this topic lecture yesterday. The uncoating can also be inhibited by different heterocyclic stops, which were very useful. Looks very useful at the very beginning. Afterwards, they turned out to have several side effects, and the replication could also be inhibited by different ways. Now, as for the possible anti-COVID therapies, from the point of view of Chalcones, are those enzymes and the spike protein itself, which are coded in the genomic organization of the COVID viruses. So the catalytic enzymes, sites of these enzymes would be suitable targets for antiviral therapy because they are highly conserved, similar to the sequence corresponding to the SARS and the MERS COVID. You might know the story. The MERS viruses were identified several years ago. So this project is underway from 15, 20 years, and which was accelerated by the pandemic caused by the COVID-19. Now let's see some essentials of the Chalcones themselves. The Chalcones have got three different important units. As you can see from this biochemical pathway, this is an open chain transitional compound of synthesis of different flavonoids, like flavonone. And we can distinguish three different important parts of the compound: two aromatic rings, whose relative position is very determining, and the polar carbon-carbon double bond. And the polarization of this carbon-carbon double bond is due to the conjugated carbonate. So this unit is called the anon unit of the Chalcones as a third or second. Several reviews appeared in the last 5, 6, 10 years on the different antimicrobial, chemopreventive, anti-cancer, anti-inflammatory agents. Some of them was written by researchers working in University of Goiás, and among them I wish to highlight only those three which are highlighted in red, namely the antiviral effects of natural and synthetic chalcones, the possibility of modulation of enzymes involved activation and conjugation of electrophilic particles, for example, carcinogens, and the anti-inflammatory effect. You might know that the most typical clinical symptoms of the COVID-19 is among them you can find the inflammation. I mean the inflammation of the lung. In this respect, now we concentrate on these two. How these two effects could be inhibited by one particular set of compounds. this review, very good one, refers several compounds, several chalcone derivatives with potential antiviral activities. Now we can I'll be concentrating on the anti-COVID activity, and this anti-COVID activity of chalcones are related of their inhibitory potential of the cysteine proteinases. The cysteine proteinases are important enzymes in the SARS, the SARS virus replication, since the genome encodes two polyproteins, and these polyproteins are cleaved to different functional proteins by a papain-like cysteine protease and a chymotrypsin-like cysteine protease. And obviously, the normal function of these two proteinases are essential for the replication of the virus. This is the mechanism of the cysteine proteinases. Organ chemistry, stereochemistry, we're just concentrating on this one. So splitting the acetylamide bound, the peptide bound, needs a nucleophilic sulfur, which is deprotonated by the histidine residue, and this deprotonated nucleophilic sulfurous attack is the initial step of this hydrolytic process. So there are several covalently bound inhibitors used as SARS-CoV-2 enzymes, and if you have a closer look of them, they also have an anion unit, the polarized carbon-carbon double bond, which can attack the sulfur. So among the thioreactive compounds, besides this semi-synthetic stuff, there are a lot of already known compounds. For example, here is the disulfiram, effectively used in treatment of alcoholism, or the seleno compound epsilon, which were also introduced with great expectations 30, 40 years ago. And among the bioactive derivatives against this protease, the literature lists several cyclic chalcone derivatives. So if you have a closer look at this molecule, for example, and you mentally eliminate this unit, you can find the chalcone compound, which is linked to by a linker, can be formed a 6-membered ring derivative, a 5-membered ring derivative, or an open chain derivative, which were theoretically and experimentally effective against this virus. The other side, the inflammatory action of chalcones, among other possible pathways, is also related to the reaction of the activated carbon double bond with the thiol function of different proteins. This reaction, when the thiol is added to the activated double bond, is called Manoel reaction. So that's the process is Manoel addition, fairly effective even in non-spontaneous ways. So the two different pathways that could lead to anti-inflammatory effect is the KEAP1 and NRF2 complex and the NF-kappa-B complex. Both of them have accessible thiol functionalities. And if in a redox reaction or an alkylating reaction, you modify these free thiols, it can activate this pathway, which results in the nucleus synthesis of different antioxidant enzymes. And the other thing is that the NF-kappa B pathway, which is activated by IKK. However, if you chemically modify the free NF-kappa B thiolate, this release, the normal release of this TNF alpha, IL-6, IL-8, and INOS, for example, just to mention a few inflammatory, pro-inflammatory agents, is blocked. So, in during our experiments, we are dealing with open chain chalcones and several cyclic chalcones with differently substituted aromatic rings and the redox active ferrosanil ring. And based on these literature data, we started a systematic testing with the National Virology Laboratory, which is part of the University of Page, of different chalcones and different chalcone analogs, with the hope finding of effective compounds which probably have got dual effects. What can we do from chemical point of view? Sulfur atom is rich in electrons. It means that the electron deficiency of this beta carbon and the stability of the product, the intermediate, determines the rate of the reaction and the position of the equilibrium. So if we compare the open chain chalcones and the cyclic analogs with different ring size, we can see that the most reactive compounds against the thiol derivatives are the inganon and the open chain chalcone derivatives, you see, and the 6-membered and the 7-membered derivatives are less effective. The other physicochemical property that we could also tune by means of introducing the substituents and changing the ring size is the lipophilic character of the compound. You can see here that first of all, if you change the substituent, the log P values are changing, and if you are changing the ring size, the log P value is increasing as long as the number of carbons CH2 units are increasing. And the third aspect that we could also modify is the stereochemistry of this enone unit, which could be so-called E or so-called Z, depending on the relative position of the let's say aromatic ring in respect to the oxygen atom. This configurational change could also, as we can see, statistically modify the log P of the compound. So we are studying, and it's underway, of the reactivity of glutathione as a model thiol, and at the same time is found in very high concentration in the cytosols, and we could see that the reactivity of the compounds really determined by the substituents and on the ring size. the cyclic Chalcones. So these are our possibilities, and that's these changing we are investigating, finding, searching for some kind of relationship between the physicochemical properties and the biological actions, namely the anti-COVID action of this set of compounds. So, as a summary of my present presentation, is that the Chalcones are versatile structures from medicinal chemistry point of view. Tuning the physicochemical characteristics of the core Chalcone structure can increase the specificity and the binding to the several micromolecules by means of proper substitution of the aromatic rings. The electrophilicity of the beta carbon can be tuned, synthesis of cyclic derivatives with different ring size. Both characteristics can be properly modified, and virus proteases are promising target of the SARS-CoV, and to the best of our knowledge, the dual acting Chalcone derivatives represent a new class of antiviral therapy. Briefly, this is our most important knowledge about this topic now, and thank you very much for the attention.
Thank you very much, Professor Paul. Very interesting lecture. We have some questions from our YouTube channel. Let me see.
Just a minute, I will try to get back to the...
Okay, Professor James is saying,
okay.
Thanks.
I'm here again.
Okay. Professor James is saying, thanks a lot for the brilliant presentation, Paul. This is a very important contribution as the world is witnessing the first Britain-approved Merck's COVID-19 pill. It's a comment. And he's asking, do you consider anti-COVID-19 screening of Chalcone analogues interesting?
Excuse me, may I just close the window, since I did not exactly hear your question. I'm very sorry.
Okay, I'm going to say again.
Sorry, thank you.
No problem, Professor.
Could you please repeat the question, please?
Yes, I'm going to read again, no problem. Professor James is asking, do you consider anti-COVID-19 screening of Chalcone analogues interesting?
Of course. Anyway, we wouldn't do that. Actually, the screening is not our job. So I don't know whether it's interesting or not. I mean, the approach is challenging to the best of our knowledge. So what we are knowing now, with cooperation with the theoreticians, is to find the overlapping structural features of the anti-inflammatory Chalcones and the antiviral Chalcones with the same mechanism of action, namely on the same thiol reactivity. This is a work at the moment in progress with some of your colleagues and some colleagues at Uni Evangelica. I think you know Dr. Hamilton and his colleagues. So no, this is a joint project between us, and we are just synthesizing derivatives of NextCat and give them to the National Virology Laboratory for testing, and at the same time we are accumulating the knowledge from the theoreticians and we are listening to the advice how to modify the structure, since it's obvious that the chalcone core should be decorated to get specificity.
Yes. Professor Jacqueline Alves, thanks for your nice talk. She's asking, has your group been studying other secondary metabolites with the potential to inhibit coronavirus replication?
Unfortunately, not really. So actually, this Chalcone project is originated of my, let's say, PhD studies, so very old. And at the beginning, honestly speaking, I was by tutoring of my older professors on the stereochemistry and the mechanism of the thiol addition on activated double bonds, which is an important feature from theoretical point of view as well. We continue this afterwards with studying the reaction with the spontaneous and the glutathione transferase catalyzed reaction of the compounds in different in vitro and in vivo conditions. And this project is a relatively new one. It's this reaction of the chalcones with protein thiols. So as you can see, this is a kind of line of development of my and my colleagues' research. And now, having the HPSCMS facility in our laboratory, we can we would find interesting to analyze, and we investigate how the chalcones react with these protein thiols. What are the structural and the kinetic and the thermodynamic feature of the reaction of this compounds with these protein thiols. So just to answer the question, unfortunately not. We are just dealing with chalcones. But in the faculty, there is a pharmacological research group who are dealing with other flavonoids than chalcones.
Okay. Ericles Mesquita, he thanks for the lecture. He's saying it was very interesting. He says, I'd like to ask if chalcone derivatives have any antioxidant pathways that could inhibit the coronavirus replication.
Yes. The hydroxylated chalcones in particular, among the hydroxylated chalcones in particular, there are very effective antioxidants. But this antioxidant effect is, how to say, it's rather non-specific, since if you apply with different pathways, there are, of course, the pharmaceutical technology can solve very, can provide very nice solution to targeted different tissues, different organs in this respect. However, the antioxidant effect is, how to say, you are biologist, you know, generation of so antioxidant effect is a very wide term. This hydroxylated chalcones are radical scavengers typically. So that's the main biological effect of its. No, that's the main molecular mechanism of their antioxidant effects. Other antioxidant effects can be resulted in very much different ways. So if Dr. Mesquita is thinking on the hydroxyl or radical scavenging effect of hydroxylated carbons, I can tell that yes, they are fairly, among them there are fairly effective ones. However, formation of these reactive oxygen species is somehow down in the mechanism which leads to oxidative stress. So this is so if you go up in the mechanistic pathway, we guess we have a better solution to prevent the coronavirus caused inflammation in the lung.
Okay, Professor Paul. Again, unfortunately, we don't have time for more questions because we have to.
Okay, no problem. We can continue next time.
Yes. Thank you again, Professor, for being so kind and for having with us during our meeting. Thank you again.
Thank you so much. Thank you very much for the invitation, and I send my best regards to all the colleagues who I'm familiar with at your university.
Thank you, Paul. Thank you. I also would like to thank all our invited speakers of this session. Thank you very much. So now we are going to have a break for lunch, and we'll be back soon for the next section at 2 p.m. Okay, so thank you for your attention. Thank you, Professor Jacqueline. So see you soon. See you.